From Microbe TV, this is Office Hours for Wednesday, the 9th of July, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where you can learn all about viruses and probably many other things as well. We give you information and scientific truths, no lies, no misinformation. Welcome, everybody. Good to see you here again. Let's welcome our mods. Tell us where you're from. Give you a shout out. We have Vanity Nutrition here tonight. We have Les Fabi and Tom Steinberg. Thank you all for coming and moderating. Appreciate it. Dobroj noci. There you go. Dobroj noci. Privyet, everyone. So, as you heard, Vanity Nutrition is moving to Montana soon. Gonna grow up a crop of dental floss. That just reminded me of the Frank Zappa lyrics. Immediately when she said, "I'm moving to Montana." Congratulations and good luck, Vanity. Hope you'll have time to join us. Probably not. You'll be busy. But wish you all the best. Don't forget, you learned it here. Moving to Montana soon. I might be moving to Montana soon just to raise me up a flop of dental floss. Raising it up, waxing it down in a little white box that I can sell uptown. I still love those lyrics. Love the lyrics. Hello, Patricia. Massachusetts finally cooled off. Here it's 30 C and cloudy. Just got back from Princeton today, where we were working on the next edition of the Virology textbook. And speaking of textbooks, I hear Quistela got your books. You got volumes 1 and 2 of Principles of Virology, autographed, and also Parasitic Diseases, autographed by Daniel Griffin and Dixon DePommier. That's a rare one because there ain't going to be no more Dixon autographs, as you know. So enjoy. Will is in Changsha with clear blue skies, 37C. Les says, amazing memory for lyrics. When I hear words, they just, boom, it's still in there. It's amazing because I can't remember so many things, yet lyrics that I heard years ago. I remember listening to Frank Zappa in college, and I'm like, what is this, raising dental floss on a farm? It's so weird and cool. How can you forget that? And the song the melody just echoes in my head. In my head, that's another song. In my head. Figure that one out. Kang is in Chicago. 22C with some clouds. There you go, Questella. I got books. Use them well. Dagfin is in Oslo. 2 a.m. 13C. Expecting 27 tomorrow. No, it's not me who's moving from New York. It's Vanity Nutrition is moving to Montana soon. Yippee-i-o-ki-yay. I'm not going anywhere. I'm right here at the incubator in New York City, although here I'm not at the incubator today. I do these live streams at home because by the time they're over, it's very hard to get back from the city. The trains are very infrequent, and if you miss one, you have to wait an hour, and the Ubers are ridiculously expensive. So I do it here. Kang tells us there's a distinction between misinformation and disinformation. TWIV is an antidote to disinformation, countering its ability to misinform us and making us more misanthropic. Very good, Kang. I like that. Christella stayed up all night reading about fleas and lice and other things. Yeah, parasitic diseases is cool. And by the way, the 8th edition is out. I believe it's on the website. You can download it for free. That's one of the things that Daniel does with his Parasites Without Borders. Gives away copies of the text. Well, he also sells them for people who want a hard copy. They're not very expensive, but you can download it and look at it. Martha's in Charleston, South Carolina, 28C and humid. There is a lot of humidity in the air tonight. Yes, totally. John is in the Twin Cities. So, John, get this. 2026, that would be next year, ASV is going to be in Minneapolis, returning to Minneapolis. I was there for ASV a number of years ago. It's a good venue. I like it. Hopefully, I can go and do another TWIV and meet you. That would be fun. So, Vanity, next week, I think I'll be driving across the country to school. Montana. Martin, it's been warm in Tacoma, but overcast and a few bits of rain really help. He has no idea that Vincent was a fan of Frank Zappa. Who next? Tom Waits. Whoever song I have listened to and I remember the lyrics, that's it. I mean, not necessarily a fan of everyone. Tom Waits would be an interesting one, right? A little eclectic there. But Jill, I love that you're a Zappa guy. Since I was in college, man, so long, totally. Brilliant musician. Kaip Tavodiena. I don't know what that is. Sveika Vanity Kaip Tavodiena. Someone translate. Rita, it's 35C in Idaho's northern Great Basin. I can hear a thunderstorm coming from the canyon lands nearby. And Mark's Tacoma is 21C. Mark Martin. Is Tom in the Oregon Coast Range, sunny, warm, and dry. Jan is back in the US of A, where it is a scorching 43C 109F in Tucson. Now, I found out where the city was, where it was 54C last week. Remember we were talking about? And I forgot it. Doha. I think it was Doha. Is that possible? Yeah, the capital of Qatar. There you go. Doha. It was 50-something, 51C there last week. Hello, Veronika. Dobroy nochi, privyet, everyone from a hot Moscow night. 23C right now. 33C during the day. Air conditioner, fan, and humidifier on nonstop. Elizabeth is in a rainy but cool 23C West Virginia. Leo is in the Mendocino Coast, 61F, and foggy. Mike and Lori Dinger in Seattle, 21C and cloudy. Mark, hey Vincent, I need to find a virologist who studies nuclear polyhedrosis viruses of caterpillars. For a Halloween episode, the larvae melt. They do. I'm sure you can find a video of that, Mark. Already planning for Halloween, Mark is. Dagfin asks, are cats susceptible to hantavirus infection? Can cats get hantavirus? So, actually, sin nombre is probably what you... Yes, cats get infected, do not show symptoms, and cannot transmit it to humans. So the kitties are okay with sin nombre. Yeah. Just horrible, I know. Dean is in Wisconsin, where it's 82F or 28C. Carol, our favorite nephrologist, is in Bakersfield, 96F, 35.5C. By the way, I didn't tell you what's up for tonight. A little bit of news, just 2 stories and a mini lecture. It turns out we didn't finish the virology course. There are several lectures that have not been done. So today we are going to look at reverse transcription and integration. What is that? Oh, it's very cool. Just stay tuned. It's very, very, very cool. Pete Glass is in London, expecting a few days of 30 to 31, but going down to Dorset tomorrow where it'll be a little cooler for 2 days. Joel, hello. Professor Vincent, thanks for your lectures. You're welcome. Logotic is in Chicago, 21C. Questella is very happy to get the books. Our pleasure at Microbe TV. EHS Talent, 25C in East Pennsylvania and thunderstorms. Hello, Luna. No. Well, that's your nickname. Virus is Luna. Luna is your cat. Hello from very warm Iowa. Laura's in Washington, PA, 23C and a bit cloudy. Thank you, my science family. There you go. This is your science family. I cried a little bit, then stayed up all night. Why'd you cry? Because you got the books? Oh, I'm sorry to make you cry, but I'm glad you like it. Tom, music and memory, discuss. I don't know anything about it except I remember songs and lyrics very distinctly. I don't know why. I forget things that are spoken to me or that I read, but when I hear things in a song, I remember it, especially if it's moving to Montana soon. You know, it's very interesting, Tom. Maybe there's a science about that, right? Joel is in Bath, UK. Very hot heat wave for 2 days. I've been to Bath. Yeah, I visited the baths. I was at a meeting, and you'll know where this is, at Cirencester. It was a very interesting meeting. I stayed in the. It was at the university, and I stayed in the university rooms made of stone, and in the shower you had to put money in to get it to run. Right, so I go in there. I don't have any money, so I had to go find some coin of the realm, put it in. Then, of course, it turns off, and it was chilly. I remember that. This is like 20 years ago. I remember that. Cheryl is in Santa Rosa, California, sunny and 76 at 5 p.m. Oh my gosh. Wait till I get to Mark's. MN, storms are moving through the D.C. area. Yeah, we have a lot of storms this summer. So last night in Princeton, as we were driving to dinner, it just ripped a huge rainstorm, lots of trees coming down and that kind of thing. Mark Martin, Dominus vobiscum et cum spiritu tuo. Oh my gosh, from the Mass, from the Latin Mass, the Roman Catholic Mass, when they used to do it in Latin. And I was a kid, my parents made me go to church. And so I just remember hearing all the Latin. And it had a mystical quality to it. And then they changed it to English. Not as mystical anymore. But as soon as I went to college, goodbye. Nobody to tell me what to do. Dominus vobiscum et cum spiritu tuo. God, I remember those words. Should I pull up some more words from the Latin Mass? Let's see. Mark, you're just jogging my memory there. The pre-1970 form. Yeah. Let's see if I can find some lyrics. We'll search for lyrics, even though they're not lyrics, right? Text for the Latin Mass. Pater Noster, Kyrie, Gloria. Oh my God, the Agnus Dei. Ay. It's too complicated to find the Latin masses. The Mass in Latin. I have an interesting book here. It's got music to it. Wow. The penitential rite, the Kyrie. Oh, it's where you say, Lord have mercy, Lord have mercy. Christ have mercy, Christ have mercy, Lord have mercy, Lord have mercy in Latin. And I remember that. You'd say it back and forth. Oh, gosh. I'll have to find these and do it sometime. Okay, back to business here. Viruses, Luna. Are you moving from New York? No, I'm not. That was vanity that I was talking about. Montana. I'm not moving anywhere. Where am I going to go? I mean, I like New York. It's a good state. I don't live in New York, but I work there and I like it. I saw today an interesting statistic. Do you know what state in the U.S. has the highest number of natural disasters? Take a guess. And what would be number 2? Hans is in Ecuador, where it's 28. And welcome back. In my head. Let's see. In my head. It's not Ariana Grande. That's not the one I'm thinking of. In my head by Zombie. Yeah, the Cranberries. That's right, Zombie. You got it. Veronica got it. Zombie by the Cranberries. Yeah. That's the only part that came into my head, because we're talking about music and memory. See how things work in the world? Just let your mind free and amazing things will happen. Last week, so we did a paper on TWIV last week about, was it 2 weeks ago? The TCA cycle and metabolism and the giant virus that has a proton gradient or some kind of gradient, electrochemical gradient. And I said, I had encountered that at a textbook meeting a couple of weeks before. And I said, I'm going to do this on TWIV. And then a few days before TWIV, I'm like, what was the paper I'm going to do? I didn't write it down anywhere. So I just sat there, let my mind free. I just let it go. And after a while, Krebs popped into my mind. I said, that's it, the Krebs cycle. So you should just let your mind run. Don't try and control it. It's good. It's good. I totally believe in that. And things come to you that you've forgotten. In my head. Okay, thank you, Veronica, for that. Jonathan is an Encinitas, 22C, and Phage Stock Prep Protocol Optimization Day. Yes, Zombie by the Cranberries. Beautiful. Peter is in Boulder, 36C, but no zircon encrusted tweezers here. No, you don't grow dental floss in Boulder. Nope. Jill is in Wilmington. Flash flood warning and severe thunderstorm warning. Our cell phones just got an EMS alert. We're safe. So here's a funny story. Just talking about EMS alerts, and just popped into my head. I was at a symposium, I don't know, maybe 2 years ago for Ian Lipkin, and it was being held in a Columbia University building. And we were sitting there listening to a talk, and all of a sudden the chandeliers shake, and I heard a rumbling. I go, oh, the subway must be right under us. It was a little earthquake, and then all these cell phones of everyone in the room started going off, and they went off in waves, because that's how they do it, right? And you could hear the sounds, and Lori Garrett, she gets up, she says, turn your phones off, because that's the way she is. Very funny. Okay, sorry about that. I forgot the new textbook smell. Yes, you got it. Dion is in the Bronx. Yeah, it's pretty heat. Pretty heat. Viruses Luna is ready to retire. You're so young. Come on. Don't ever retire. Always use your brain. I am not going to retire until they carry me out on a stretcher. Tom from Vanity. I can't recall where I learned this, but I heard a retirement home started playing music popular when their residents were young adults. It had a positive effect on them. Not surprising. Not surprising at all. You know, I remember today's music less. Well, I don't listen as much as I used to, right? Virus is Luna. I could go to Minneapolis. It is close. Yeah, you could. Or I could come to wherever you are. We'll talk. Brian is in Bridgeport, Connecticut. Man, weather's probably the same as where you are. Elena, privyet from University of Nevada, Reno. We've been having a mild summer so far. 83F today. About to heat up in the next few days. No humidity whatsoever. Whatsoever. Forever and ever. Oh, wait, what's that from? And it's just the way they said the accent. Forever and ever. Okay. John is in Minneapolis, of course, and he would go to ASV. Yeah. Come. ASV is a little pricey, right? But we can certainly meet up for sure, and whoever else is in that area. So for ASV next week, which is in Montreal, we're having a meetup. If you're going to the meeting, we're having a meetup on Wednesday evening from 9 to 10 p.m. in room 503, I think. Probably nobody here is going to ASV, I guess. Jeff is in Elgin. We're at 76F. Misinformation and disinformation are lies. Ripticomp, Stinkfoot from Taos. Okay. Maureen is in northeastern Ohio, 79F with light rain. Pete Glass, Zappa's Hot Rats album, a total classic. A young band did a night celebrating it a couple of winters ago at the Jazz Cafe in Camden Town. I know where Camden Town is. I've seen this the tube stop. Yeah. Joel, thank you for your support of science communication. Come on, everyone, pay the Twiv gang some money for all the great content. Yeah, we don't charge you for this content. It's always my plan to give it away. For a while we had ads, and then I just stopped that because I feel it's disruptive. And I said, let's try and run on people's support. And it was okay. But then when COVID hit, boom, then we had support big time. And that's when we made Microbe TV a nonprofit. So we wouldn't pay taxes, right? And so all of the money could go to our operations. So that's the history. Eliza B., 25C and thunderstorms in southern Ontario. Happy-go-lucky. We're planning to take our baby to Vietnam next February. 2 weeks after his early MMR shot at 11 months. With the measles outbreak there, do you think that timing is enough? Well, yeah, 2 weeks is okay, but you know, there's another dose later. Obviously, you can't get it, but this is better than nothing. Yeah, you have to be careful, right? That's the best thing you can do is 2 weeks. Yeah, Vietnam is apparently amazing, beautiful, great food. For some reason, I'm getting all these Instagram posts about Vietnam. It's incredible. You're going to have a good time. I'd love to go. Okay. Whenever Mark Martin mentions Tacoma, that makes me think of Galloping Gertie. Polka Dot Pup, good evening from North Texas, where it's 31C. Rima. Sorry. When I volunteered at our local elementary school, the teacher and I would trap lice with packing tape. Yikes, what I learned in kindergarten. Wow. Virus is Luna. HIV is her favorite virus. Javier from Southern Ontario, 26C and raining. Rima. Vincent, you forgot. But you still remember Luna. Yeah, how could I forget Luna? So I have a bad memory for names, but you haven't been around a lot. And we've been chatting less on Instagram than we used to. So that's why your name slipped away. I was trying to get it before, but obviously I couldn't. Sorry. Dr. Schonbaum, a resident dentist. Sunny and clear at the beach in El Segundo. What's with the new policy at NIH on lab animals? They want you to use fewer lab animals and use AI to predict whatever it is you're doing would do in animals, right? But we don't have enough data yet to do that. Any thoughts on the latest research indicating that a normally harmless virus might trigger or contribute to Parkinson's disease? Virus, Parkinson's. Let's look it up. A human Peggy virus. It's been found in the brains of individuals with Parkinson's, but not in control brains. So it could be a factor. All right, so I don't know if that's the one. Common virus could trigger Parkinson's. There you go. All the news studies. Peggy viruses. Flaviviruses. Related to hepatitis C, distantly related. Now, it's always hard to know because many people have this virus, and so it could be just an association. So obviously we need to do more work, look at more patients, but it's interesting, right? But maybe there's something else that makes it a risk factor. We'll have to look at that paper and see if it's twivable. Thank you for the Q. Clue. No, Q. 110 people, 67 likes. Hit the like button, folks, to attract more people to real science here. So Les asks Rima if Luna the cat collects viruses, and viruses Luna has infected me with cat love virus. Just makes you adore viruses. Veronica says songs probably engage more parts of the brain, memory, easier to remember. F. Yang is in Houston, where it is 28C. No worries about being late. It's only 27 minutes in. No problem. Joseph is in Ontario, Canada. John is in Port Townsend, Washington, the cool northwest part of the lower 48, where it is 57. Welcome back, John. And Les tells us, before books, information was saved by singing and making lines rhyme. It's a good way to help you remember things when you have a rhyme to it, right? Hello, Noir. Welcome from wherever you are. This is a poet, right? Hello, Noir. Welcome from wherever you are. Now, you may have heard, did you hear yet? No. So on the next Beyond the Noise, at the end, you're going to hear me say, what they're doing is MAGA, Make America Germany Again. And that's courtesy of Noir. I didn't say it on the show, but now you all know Noir made that up. Thank you, Noir. Kiri is Greek. Dagfin says, et in terra pax hominibus bone voluntatis. What's it mean? I don't know if this one is doable. Let's see. Yeah, it's been deleted. We will not do that one. Were they all in Latin before 1970? I just remember, according to Wikipedia, yeah, the masses were in Latin before 1970. I remember growing up hearing the priests do the Latin droning. It was kind of mysterious, as I said. No. The state with the most natural disasters is actually Texas. Yeah. Is that interesting? Let me look it up. Texas and California are the two top. Texas with the hurricanes, tornadoes, floods, wildfire droughts. California, earthquakes, wildfires, and floods. Yeah. Florida's pretty high. Louisiana, Oklahoma, Mississippi. New York, Hurricane Sandy. Okay. Here's another one I'm not going to answer. Dagfin, Latin was exchanged for local languages after the Second Council of the Vatican in 62-64. Thank you very much. John, it was Pope Paul, right? Was it Paul? Pope John the 23rd. So he was the Pope when I was a young kid, and he was pretty progressive. And he had this Vatican Council, encouraged them to convert it to local languages. Yeah. Cranberries in your head. Greeks were afraid that books would weaken and cripple human memory. Well, got lots of books here next to me. Not crippling me. Tons of books. Jesus in Seoul, 30.8 C and will rise to 36. Welcome. I don't remember having anyone from South Korea. Welcome, welcome, welcome. Country I always wanted to visit. Steen, hello there, Mr. Plant, Dr. Plant Virus Man. How have you been? Looks like ASV was previously in Montreal. Did you go? I did. Any memories? It was good. Montreal is a good city. It was very hot because it's summer, right? But that's typical. But Montreal, there's old Montreal, which is very interesting and has nice character, nice restaurants. It's a good city. So if you're going, you'll like it. Yep. Do you have to let it linger? Really? Do you have to let it linger? Oh, this is just a great song. It's a great song. Thank you. Mark says, Vincent, you can hear music and singing in your mind, right? I can. I mean, I can hear the song just as it was performed when I heard it. Peter says, Zombie is outstanding, played at volume 12. Oh, gosh. This is memory lane. And the lady, this lead singer, she died, didn't she? Unfortunately. Zombie. Okay. We won't go there. Thank you for all the information. Vanity likes the cranberries. Yes, they're good. Marco is in the east end of Long Island, where it is 25C, not enjoying these humid days. Spamlet, 26C, 84% humidity in my Bedfordshire kitchen, where I've just released some expensive Aphidoletes, Aphidimisia, or aphid midges to try and rescue my greenfly infested chili plants. Biocontrol. Yeah, go for it. Heather says, Cranberry Zombie is currently one of my 8-year-old's favorite songs. And Carol, you got it. Texas has the highest numbers of natural disasters. Elcio from Belém, 32C in heavy rain. Marco Mont, I think Texas has the highest natural disasters. That's right. Tryon, always with a pithy question. Why do some viruses infections trigger immune intravascular hemolysis? Well, there would be two possibilities, right? Well, viruses can't reproduce in red blood cells. So they could reproduce in the precursors and then make you have fewer red blood cells, but that's not what you're asking. But I'm suspecting it is a inflammatory reaction, right? So depending on the kind of inflammatory response a virus elicits, it could trigger this kind of hemolysis. Yeah. I know, forever and ever just popped into my head. Let's look. Let's see if I can find it. That's a tough one. Some songs are popping around in my head, but I don't know what they are. I don't know. I'm stumped on that one. Mark is in Las Vegas at 110F, 43C, no hope of rain. Oh my gosh, that's tough. John, I would wash my hands and cook food for any meetup I could be part of. We'll do something cool. Mar, thank you for your contribution to science communication. We really appreciate it, and smash that like button. We only have 85 likes and 125 people. Why do I want you to smash the like button? Because it could attract other people to see what we're talking about, you know? Manity says, music activates the area of the brain involved in emotions and controlling memory, the limbic system. Music is wonderful, isn't it? Joel is going to sleep. It's 1 a.m. Watch it tomorrow. Thanks for coming by, Joel. SRR is in Paso Robles, where the temp is 306 degrees Kelvin. Jeff, I never listened to Zappa for whatever reason, but found the documentary Zappa very interesting. The guy was near genius, if a genius. Yeah. Very Nika says it would be cool to have a guest from Twin or TWIV for papers on viruses being associated with neurological diseases. Yeah, for sure. I'm very... So on Twin, when it's my turn to do a paper, I'll typically do that. But having a guest is great. I agree. We're having guests Friday on TWIV. Talk about RNA recombination in enteroviruses. Vanity did some COVID carols and one for Mpox. So the virus is still monkeypox virus. The disease is Mpox. The International Congress for the Taxonomy of Viruses controls, regulates virus naming, and they have said it's still monkeypox virus. And the WHO changed the name of the disease to Mpox. And also, an opera singer from Santa Fe and her husband, she sang and he played the piano. They did 2 COVID songs for us. You can find them on our Micro TV YouTube channel. Just search for the COVID song. Actually quite good. Markle, I'm holding my breath. COVID seems to be at a low point. Yeah, so I said to Daniel weeks ago, I think we're not going to have a summer surge like we did last year. And I have no data to make that conclusion. It's just that it's weird that respiratory viruses surging in both the winter and the summer. So typically influenza in temperate climates surges in the winter and then very, very low levels in the summer. So I thought maybe SARS-CoV-2 had to do a couple of cycles of surge to get population immunity maybe to the point. And so far there's been no surge. But we'll see, as Markle says. Yep. Steph SF, our moderator, has joined. Welcome. Sandra says it means peace on earth and goodwill to all humans. Okay, thank you. Hello, Sandra. It's okay. You wanted to explain it. It's a good teacher thing to do. Very Nika. The only Zappa song I have on my playlist is called Why Does It Hurt When I Pee? I guess he was really good at coming up with such lyrics. Yeah, he liked to shock people, right? He had an album called Weasels Ripped My Flesh. You're going to Make America Germany again. But I said Noir's thing was Make America Germy. Germy. I don't particularly like the word germs. It lumps bacteria and fungi and viruses together. But people like to use it, and it fits for MAGA, right? Here we go, Dagfin. And peace on earth for people of goodwill from the glorious sequence of the Tridentine Mass. Yeah. John writes, discussing vanity songs is making me realize I've been watching about 3 years of Office Hours Q&A. This channel made me think of my health and quit smoking. Good for you. It's great, John. It's not good to smoke. And, a lot of kids are vaping these days before we really know what are going to be the long-term consequences, right? I don't like it. Florida is the lightning capital of the nation. Viruses Luna says, very interesting that Luna understands when I talk to her in all the 5 languages I know. What are your 5 languages? You got English and I guess Lebanese, right? What else? What are the other 3? Tell us. I'm curious. Spamlet now. Oh, Stevie Ray Vaughan. Great cover of The Sky is Crying. Oh my gosh. It just makes shivers go up and down my spine. The Sky is Crying. It was, remember, Jimi Hendrix did that. Very good. But Stevie Ray Vaughan is amazing because he's an incredible guitarist. But Spamlet's remembering Texas flood. So sad. YouTube of Vaughan and Albert King jamming the old blues standards had me in floods of tears for all the lost souls, including poor Vaughan and King. And Vaughan was killed, right, in a plane crash, I believe. P-Glass talking of Parkinson's, also a link suggesting Shingrix vaccine is protective against Alzheimer's. Nature. Yes. Interesting. Have to look at that too. There have been some cases where Alzheimer's has been transmitted by giving people pituitary growth hormone. It was contaminated with misfolded protein. So all these neurodegenerative diseases could all be misfolding protein diseases, like prion diseases of misfolded proteins. Maybe Alzheimer's and Parkinson's, they're all the same. Very cool. My mom's uncle says Red Hen 689 got flu during 1918. I have heard he developed Parkinson's disease as a result, but others called it encephalitis. Well, influenza viruses. They don't cause encephalitis typically, right? So I don't know what that, maybe it was something else, right? Remember Occam's, what was it? Occam's razor and Hickham's dictum. You can have as many infections as you damn well please. Okay, sorry about that. Marsh, do immunocompromised people need to get a measles booster? No, you shouldn't, because they shouldn't, because measles is an infectious vaccine, and you do not want to give infectious vaccines to people with immunocompromise of any sort. Yeah, unfortunately, we don't have an inactivated measles vaccine. F. Yang says Texas is horrible at this time. For what you don't pay for taxes, you pay for fixing stuff and food spoiling in the fridge for a week-long power outage. Jeff writes, as I remembered, the Mass changed around the time when I was in third or fourth grade. In Berwyn, the Mass was in Latin and then English when I went to St. Hubert's in Hoffman East. This Greek isn't afraid of books. Veronica, Texas. Always wanted to join those tornado hunters. Also, I think USA has vastly more tornadoes than anywhere else in the world. Let's look it up. Who has the most tornadoes? The U.S. Between 1,000 and 1,200 tornadoes every year. That's not a distinction that you want, right? Twiff and fungus? No, I don't think so. Not enough, I don't know, interest. That we do fungi on TWIM, microbiology. I think that's good. People have asked for this for years, but I never wanted to do it. Kang says Daniel Dennett distinguishes disinformation as giving misleading information, and if someone accepts, he becomes misinformed. All right, so that's true. Disinformation is a conscious effort to mislead you, whereas misinformation is you're just wrong. You're just wrong. So is RFK misinformed or just lying? Well, it's probably both. He's misinformed about a lot of things, and he lies about a lot of things. He says the measles vaccine kills people. No, it does not kill people. I know one person who was killed, and they were immunocompromised. So, Noir, there you go. An immunocompromised child got measles vaccine, and it killed him because it's an infectious vaccine. Vanity writes, there's a strange relationship with Parkinson's and melanoma. Parkinson's patients are much more likely to get malignant melanoma. I don't know what's involved there. I don't. Hello, Coralie from New South Wales. Welcome to Office Hours. Vanity, for after the stream, here's the link for the video for Zombie. Spamlet writes, I wasn't religious, but sung Latin songs in church choir as a child. It taught me that most songs are awful when you understand the words. Can't sing the good ones because they make me cry. Interesting. Virus says, Luna, I played the piano for 5 years, and then I decided to stop because I sucked all my senses and it absorbed me. Science and athletics are better. I don't know. I think playing an instrument is amazing. I have one back here. I don't know if you can see it. Here, it's a guitar. Well, let me get this off. Maybe you can see it. Yeah, you can just see the handle. I used to play that, and I used to play clarinet. It's very fulfilling. But what I'd really like to do is play the piano, but that's not happening. I mean, I have one right above me. My mother's baby grand, but I don't have time. I'm doing other things here at Microbe TV. Spamlet, same here, Vincent. I rarely play records. I just have to think of the cover and a few notes soon get me going to hum along in my head. I have a big vinyl collection, but I don't play it much. I have a big CD collection from the old days, but I don't play them much. What do I do? Yeah, I go to YouTube and look it up and play it if I want to really hear it. John, thank you for your support of science communication here at Microbe TV. We need your help. Really appreciate it. 132 people, 101 likes. Please hit the like button. Very Nika, forever and ever, you stay in my heart and I love you comes to mind. I don't know what it is. It'll come in. I don't want to take the time now. Mark Martin, thank you for your contribution to Microbe TV. I really appreciate it. Vincent, didn't they make you take Latin in high school? The good Javierian brothers spent 4 years trying to bang it into my head and failed. They took a couple of years in college and loved it. No, they didn't make me take college in Latin in high school. No. I didn't go to a Catholic high school. Maybe that was it. I used to be able to work listening to spoken words, but could not concentrate if music was playing. I can't work listening to spoken words. I get distracted, but I can work listening to music. Yeah. Patrick is in Puget Sound, Washington, 72F. Perfect. Perfect. Yes, music is wonderful. Veronica said that's by Aretha Franklin. Say a little prayer. Conrad, hey, Vincent, you really helped me through the pandemic with great information. Sad there's still so much vaccine misinformation out there, especially COVID-19. Oh, dear. There are people who get wealthy by peddling vaccine misinformation, disinformation. And now the country is, the science and public health agencies are being run by people who don't like vaccines. They have power now. It's terrible. It's totally terrible. Anyway, I'm glad you enjoy it. Javier said, I got to hear a stadium full of Irish rugby fans sing Zombie upon beating the South African Springboks, absolutely electric atmosphere. I'm sure it was. Mark, have a good evening. Thanks for joining us. PMFG, thanks for providing a virology channel on YouTube. You bet. You bet. So Tryon explains cold agglutinin can induce erythrocyte aggregation at peripheral capillary circulation, and erythrocyte after aggregation can be destroyed at that level. Okay. Forever and ever you'll stay in my heart. Carol says the American Academy of Pediatrics is suing HHS over their vaccine policies and put an end to the assault on science and evidence-based medicine. Well, it's funny you should mention that. That's one of our news stories tonight, so let's do it. So we have, let me open the story here because I downloaded it from the New York Times so you can all see it. Here you go. This is by Apurva Mandavili. Medical societies sue Kennedy and HHS over vaccine advice. New restriction on COVID shots run counter to scientific advice, the groups say. 6 leading medical organizations filed a lawsuit in Western Massachusetts. Seeks to restore COVID vaccines to the list of recommended immunizations for healthy children and pregnant women. Mr. Kennedy has been on a decades-long mission to undermine vaccines and portray them as more dangerous than the illnesses they're designed to prevent. The secretary's intentions are clear, Mr. Hughes said. He aims to destroy vaccines. Plaintiffs include the American Public Health Association, the American Academy of Pediatrics, the Infectious Diseases Society of America, the American College of Physicians, the Society for Maternal Fetal Medicine, and the Massachusetts Public Health Alliance. They are joined by a pregnant woman who said she was unable to get a COVID shot. Mr. Kennedy's decision contravenes years of evidence showing that pregnant women are higher risk of severe illness, miscarriage, and stillbirth if they contract COVID. But from a scientific perspective, a pregnant woman by her very nature is immunocompromised. Mr. Kennedy's decision didn't follow the normal processes. So this is great, and the article continues, and it's worth looking at. But I don't know what's going to happen. Any lawyers out there who can tell us, like, what'll happen? So who's going to decide this suit? And then if they find him guilty or whatever it is that the outcome is, what can they do? Because, Trump will tell him, don't worry about it, just ignore it. And would they arrest him and put him in jail? I mean, I don't know what the outcome would be. I mean, it's good. It gives more publicity to this, but I just don't know what's going to happen. Okay, so that was one of our news stories. And then the other one here, this is a good one also. From Science, I will put this up here. It's a nightmare. U.S. funding cuts threaten academic science jobs at all levels. So this is about University of Maryland professor Ann Simon, who's a plant virologist. And she had an assistant research scientist. She told Xiao Bao that she couldn't extend his position as they had originally planned because the U.S. Department of Agriculture, who had agreed to fund a trial, took it back. Very hard because you're devastating people's lives, Simon said. I never had to do this in 38 years. Ying, a single father who has worked in Simon's lab for 5 years, is now scrambling to find another research job. He's only seen one position he feels qualified for. In the meantime, he plans to get by as an Uber driver. So this is going on in many laboratories. And there was a number here that I thought was good, if I can find it. So here's Ann Simon, who I have danced with at ASV. And there is Xiao Bao. Look at the plants. Aren't they great? Where's the numbers? Where are the numbers? We talk also about restrictions on visas and so forth. Oh, I can't find it, but basically the number of postdocs and PhD students are going to be cut. Here we go. All right. So if U.S. academic science as a whole, numbers released in May, the number of early career researchers it supports, the NSF, could fall from 95,700 undergraduates, graduates, and postdocs to 21,400. It's a nightmare, Simon says. I really feel for the future of science. NSF declined to comment. Chickens. Okay, so those are our two stories. The onslaught against science continues. So that was from Carol. So where is Carol's chat so I can resume? Here we go, Carol. Yeah, but Handel's Messiah has a forever chorus also. Maybe that's it. I don't know. Where is Laura's comment? It just pops all around now. What's going on? Here we go. Michael Schmidt would be my pick for a guest. On what? On office hours? Yeah, he's been here before. If you want him back, we can do that. We can do it. Polka Dot Pug. I love to watch the music videos on YouTube. I watch EDM, the DJs spinning. I love it because they go to weird places, interesting places. I love it. Dagfin, I believe I wrote the lyrics for one of those COVID songs. Which one? Someone should ask Michael Schmidt, very Nika says, if by any chance he knows what dark matter is made of, because he seems to know everything. He does, doesn't he? It would be interesting. Tomorrow's Thursday. Oh my gosh. TWIM. I don't know if anyone's got papers together. Mr. Labman. Okay, Dagfin wrote the lyrics to Mr. Labman. So you know the people in Santa Fe, the opera people, really? That's cool. Let's see. Michael Schmidt. F. Yang remembers seeing Cameron and Bohr's proposed polio 3D pole recombination mechanism. Yeah, well, we'll see. So I'll have David Barton and Ulf Pearson on to talk about recombination. Will says SARS-CoV-2 is a vascular disease that starts in the lung. I don't think it's a vascular disease. It's a respiratory disease that starts in the upper respiratory tract. Okay, that's where the virus comes in. It reproduces in the nasopharynx and then spreads down into the lungs, and it reproduces in lung cells, alveolar cells. Okay, the endothelium, I was never convinced of it. Okay. When I go on Mark Thompson, try to explain to him about lab animals. He had a rather one-sided discussion with an animal rights guest yesterday. Okay, yeah, that's okay. Camera on, no problem. Interesting. Who the guest with enteroviruses is. So it's David Barton, Brian Kempf, his associate, and Ulf Pearson. They're all at the University of Colorado. Okay, viruses Luna, microbe TV. Oops, sorry. Lebanese, English, French, German, Bosnian. Wow, that's amazing. Bosnian. Abdur Rahman, welcome. No problem being late. Bosnian, yes, so some Russian too. Privyet. Anybody know that the great Beethoven pianist Alfred Brendel passed away 2 weeks at age 94? Did you know that Franklin Stahl passed away in April at 96? Do you know who Franklin Stahl was? The Mendelssohn and Stahl experiment. Let's see. I know that Tom Steinberg knows what it is. Mendelssohn and Stahl. We're going to do the paper on an upcoming TWIV. Loss of smell is thought to be an early indicator of Parkinson's, presenting years before motor symptoms. Could that be from an initial virus infection? It could, but these PEGI viruses are not respiratory viruses, right? They're hepatitis viruses. Anyway. So pedantic objection. The ICTV changed the name of the species to orthopoxvirus monkeypox, but they do not have formal authority over virus names, so Mpox virus is valid. That's not what Jens Kuhn told me, and Jens knows it all about ICTV. I'll have to ask him. I believe you, Steen. Who told me that? I think it was Jens who told me that. Yes, it was Jens who told me that. And Jens is pretty accurate. The Oliver Sacks movie Awakening was about 1918 flu causing Parkinson's and L-Dopa treating it. Let's see. Yeah, I mean, it's an association, okay? People born during the Spanish flu had a 2 to 3-fold increased risks later of developing Parkinson's compared to those born before 1888 or after 1924. Okay, but it's an association. There may have been other factors that, in fact, increase their risk. So you have to be very careful about that. Questella, if you have 47 tornadoes in one day, is that 47 natural disasters or one? That's one, I think. That counts as one. To be fair, with respect to natural disasters, both California and Texas have the most land and people. The disaster rate would be a better metric. Rita says, I feel RFK is malign, not just ignorant, purposefully working against public health. Yeah, we call it malignant incompetence. I also think he's mean. Yeah, I do. Min, love your channel. It's my first time for office hours. I heard that Twiv will be at ASV. What would the topic be? You can't know. It's a secret. It's a surprise. I'll be there. Come. It's Thursday at noon. Come and you'll see the topic. Costello has a clarinet and a saxophone. Saxophone is pretty cool. Artist Egon Schiele died October 31st, 1918 of influenza. Yes, the infectious diseases take all kinds of people's lives, don't they? GSD Bio says maybe the susceptibility to disease prevention misinformation is related to this. Upton Sinclair. It's difficult to get a man to understand something when his salary depends on his not understanding it. I like this name, Michelozzo Michelizzi. Wonderful, Michelozzo Michelizzi. Hit the like button. Noir took Latin 1 and 2 in secular high school. Okay. Mine didn't offer it. PMFG plays the clarinet, and certain jazz songs can have an effect of mathematical symmetry, for sure. For sure. Carol says to Vanity, wishing you the best in medical school. My daughter just started her clinicals, third year. Remember to have fun. Luna is listening to Office Hours, and she understands and says hi. Luna the cat. Rita didn't know about microbe TV during COVID times. It was H5N1 that got me here. It doesn't matter. But I tell you, we had a huge uptick in listeners during COVID, starting in January 2020. And here we still are. Thanks, SRR, for your support of science communication. Really appreciate it. The daughter of my friend is studying healthcare communication. Can she have an internship at the incubator? So I was just talking to someone about this today. I would like to have an intern who shadows me and learns everything that I do and then writes it down, right? So that should anything happen to me, it can be done by others, right? There should be a protocol for everything. Right now, it's all in my head. And so I'd like an intern to, so let's chat, okay? The daughter of my friend. But Rita says, if I knew about you all during the pandemic lockdown, I would have been here then too. No problem. It's okay. You're here now. We got you. Well, physicians should join in this lawsuit. It should be a tsunami against RFK Jr. So the case is civil, so jail is inappropriate. So what can they do? Fine him? What's the point? Hello, Jessica from Toledo. Nice to see you. Ray says, suing a lawyer is tough. Good luck. Not going to win it with facts. Unfortunately, the pregnant woman can get a vaccine. That's hogwash. Might have to pay for it unless the doctor fears liability. Yeah, it's not covered by insurance, right? Hello, Margot. 47 in Phoenix. That's hot. Wow. Yep. Skulls. F. Yang got into TWIV from my undergrad professor, who was a student of the coronavirus researcher David Bryant. I never could have thought I would be working with a virus protein only 2 years later. Min, I love your channel. You're welcome. All right, one more. And then we'll do a mini lecture. Tona wants to know if shingles is transmissible. My 50-year-old brother has shingles. Wonder where he got it from and if a non-Shingrix vaccinated adult could catch it. So you don't catch shingles. You get chickenpox as a child, right? And the virus remains latent in you. And then it's reactivated when you're older, 50 years old and up, typically. And so, no, you don't transmit shingles. You transmit the chickenpox virus, which you got earlier. Is that clear? I hope that's clear. Okay, let's mark that and go to a mini lecture. This is cool, reverse transcription and integration. Okay, let's get this a little larger here. Okay, this is one of my favorite quotes from Alice in Wonderland. One can't believe in possible things, said Alice. And this now the Queen is responding. I dare say you haven't had much practice, said the Queen. Why, sometimes I've believed as many as 6 impossible things before breakfast. That's reverse transcription and integration. So a little history. So this is a feature of many viral elements, but particularly tumor viruses where it was discovered. So in 1908, and this is part of our lecture on transformation and oncogenesis. In 1908, chicken leukemia virus is discovered by Ellerman and Bang there. And 1911, Rouse sarcoma virus discovered by Peyton Rouse, who got the Nobel Prize 55 years later. So these were the first 2 cancer-causing viruses to be discovered. And they were called tumor viruses because they caused tumors. And eventually, they were found to have RNA genomes. So they were called RNA tumor viruses. And many people tried to figure out what was going on. How do these RNA viruses change cells? They transform them. They cause permanent changes in the cells they infected, which eventually became tumors. And he said, okay, this is an RNA virus. The RNA must be changed into DNA, which can then integrate into the host genome and become a permanent part of the host DNA. So he formulated, and that's Howard there, very smart man, the provirus hypothesis, which said that the RNA tumor virus DNA was integrated into the host genome, and that's called the provirus. At the same time as Temin was working on this problem, trying to understand how RNA tumor viruses transform cells, change their properties permanently, his insight was that he picked up on Temin's ideas and said, okay, how can an RNA tumor virus make DNA? He knew that plus-strand RNA viruses, there is no RNA-dependent RNA polymerase in the virus particle because the plus strand can be translated as soon as the virus gets into the cell. But minus strand viruses have to carry the RNA-dependent RNA polymerase in the particle because the minus RNA cannot be translated. And so the polymerase has to be there to make messenger RNAs from it. So he said there has to be an enzyme in these RNA tumor virus particles that copy RNA to DNA. He says it has to be in the virus particle. So he went looking for it. And in 2 weeks, he found it. And he called Howard Temin. And he said, Howard, I'm going to publish this. I know you've been working on it for 10 years, so I'm just giving you a little heads up. So Temin did the experiments very quickly. And so they both published back-to-back in Nature in the early 70s of the existence of an enzyme in these RNA tumor virus particles that can convert RNA to DNA. So it's called an RNA-dependent DNA polymerase. And Temin worked on Rous sarcoma virus, and David worked on other RNA tumor viruses. But again, the idea was this validates this whole hypothesis that the RNA tumor virus can make permanent changes to cells because its RNA genome is converted to a DNA copy, which then integrates into the host cell. So the enzyme is called reverse transcriptase. And the viruses are called retroviruses now, no longer RNA tumor viruses, because they can reverse the flow of genetic information, right? Normally, we go from DNA to RNA to protein, but reverse transcriptase can take RNA and convert it to DNA. And that DNA integrates into the host. It gets converted to mRNA, and then that's made into protein, of course. So retroviruses, that's why they're called retroviruses, not because they're retro. This enzyme has revolutionized molecular biology, and neither scientist patented it. For example, you can clone genes by taking RNA and converting it to DNA. Every RT-PCR, if you're looking for an RNA virus in a clinical specimen, for example, you use reverse transcriptase to convert it to DNA, and then you use PCR to amplify it to a detectable level. And so you can buy reverse transcriptase from a variety of companies, and it is just a gold mine for molecular biology. And so here's the Baltimore scheme showing the 7 classes of viral genomes and how they get to mRNA. And so the retroviruses have reverse transcriptase in their reproductive cycle. Of course, we just said that. They have RNA in the virus particle. It's converted to first negative DNA and then plus minus double-stranded DNA, which is integrated into the host and then it gets copied to mRNA. The hepatitis B viruses, which have partially double-stranded DNA genomes, they also have reverse transcriptase in their reproduction cycle. And we'll see that at the end. So I have to go back to David Baltimore here. So he had said there must be an enzyme in the particle to copy RNA to DNA because he said there isn't an enzyme in the host cell that can do that. Well, he was wrong. Many years later, they were found. So our cells have reverse transcriptase in them. Very low levels, though, not enough to do what the virus needs. So here's Rous sarcoma virus. This is a retrovirus. It's an envelope virus with spikes in the envelope that mediate attachment to cells. Inside is the plus-stranded RNA genome. And inside the envelope is an icosahedral particle. The genome looks like this. So Rous sarcoma virus or avian leukosis viruses. This is the proviral DNA. This is the DNA that's integrated into the host cell. So it encodes GAG protein, Pol protein, and envelope. So the GAG is the structural protein that makes up the particle. The Pol are the enzymes, including reverse transcriptase. And then the envelope, of course, is the glycoproteins that stud the membrane. And so this genome integrated into the host cell, it's flanked by what are called long terminal repeats that have promoters for the initiation of RNA synthesis. And as a provirus, it makes a long mRNA, which is translated to make the GAG proteins. And then there is a splicing event that allows synthesis of the envelope proteins. And then there's a read-through that allows synthesis of a GAG-Pol precursor that is eventually processed to give reverse transcriptase and integrase. So here's the reproduction cycle of these retroviruses. So the virus is attached to a receptor. Many of them, the membrane of the virus fuses at the plasma membrane. It's taken up into the cell, and the reverse transcription initiates in the cytosol, and a double-stranded DNA is made from the viral genome. There are actually 2 copies of viral RNA in the particle. The DNA is imported into the nucleus. It integrates into the cellular genome, and there it's transcribed to make messenger RNAs that go out into the cytoplasm and make the viral proteins that are used eventually to make new virus particles. And of course, some of these messenger RNAs end up being viral genomes incorporated into the new particles. So let's talk about reverse transcription. We're going to focus a lot on this, and then next time we'll talk about some other topics related to reverse transcriptase. So reverse transcriptase, as we said, in the retrovirus, copies RNA into DNA, and it's a primer-dependent enzyme, so it needs to have a short primer. And the primer can be DNA or RNA. And actually, the template can be RNA or DNA, as we'll see. So RNA is what the virus has in it, and it can copy that and make DNA, but it can also copy DNA, as you will see. But it only synthesizes DNA. It does not synthesize RNA. So only deoxy-NTPs are incorporated, not ribodeoxy-NTPs. So it makes only DNA, even though it can copy DNA or RNA. It turns out that our reverse transcriptases are found everywhere in bacteria, archaea, and eukaryotes. So here is a very high-level phylogenetic tree of the 3 domains of life: bacteria, archaea, and eukaryotes. And so you know all the bacteria. Archaea, of course, are ancient organisms that are not bacteria. They're distinct, and they're not eukaryotes. But they all have a common ancestor. And that common ancestor must have had reverse transcriptase, because bacteria, archaea, and eukaryotes all have reverse transcriptase. So RT must have evolved before the separation of these 3 domains of life. Now, we think that reverse transcriptase may have been the bridge between the early RNA world and the modern DNA world. There's a hypothesis that at one point on Earth, billions of years ago, there was an RNA world where organisms had only RNA. In fact, they were initially just replicating RNA molecules, and we don't know how far that got, if it even became an RNA-based cell. But at some point, it transitioned to a DNA world, and it could be that reverse transcriptase, which probably evolved early, caused that transition. Now, at the bottom here is a circular phylogenetic tree of reverse transcriptases in different organisms. So here are the virus reverse transcriptase. So hepadnaviruses have RT, retroviruses, and also colomoviruses, viruses that infect plants. And then there are a variety of elements, transposable elements that have RT, including group 2 introns. Bacteria have it, and archaea. Telomerases, the sequences at the end of our chromosomes, have RT. And then line elements that are integrated in our genome have reverse transcriptase associated with them as well. So RT is everywhere. Reverse transcriptase is one of the 4 known nucleic acid polymerases. So there are 4 polymerases. There's DNA-dependent DNA polymerase that copies DNA. There's a RNA polymerase that copies DNA and makes messenger RNA. There's reverse transcriptase, and then there's RNA-dependent RNA polymerase. And these are the structures of the proteins. They all have an active site, which is seen as these yellow beta strands here. And these polymerases take on the structure of a right hand, where the palm is the active site. And for some, the fingers and thumb touch, like in the polio polymerase, and for others, they do not. And above are the sequences shown as bars for these polymerases. And you can see there's some conserved regions in the red and green and yellow and purple. And the yellow comprises the active site. Those are the 2 beta strands that I showed you there. And there are very specific amino acids that comprise the active site for different viruses that you can see here. The HIV RT has a similar trio of amino acids in the active site. And these amino acids are important for holding magnesium ions that are an important part of catalysis for all the polymerases. Now, the reverse transcriptase has a second activity. It has RNase H. And this is an enzyme that cleaves RNA when it's in a double-stranded form or a duplex form. And the RNA can be in an RNA-RNA, could be double-stranded RNA, or it could be an RNA-DNA duplex. And what the enzyme does is make what we call endonucleolytic cleavages. They cut inside the double-stranded RNA. If it were cutting from the ends, it would be an exonuclease, but it's cutting inside. It's an endonuclease. And it chops up this duplex into short oligonucleotides. And you'll see why this is important in a moment. Here's the structure of HIV reverse transcriptase, made up of two subunits. In the middle, the two subunits are joined together. And what we're showing here is the palm domain, which contains the active site. And an RNA in green is passing through the active site, and a DNA copy of it is being made. That's in blue. So a single strand RNA comes in. The enzyme, it's copied into a DNA, and then you have a DNA RNA double-stranded molecule. Here's another cartoon of the reverse transcriptase showing what I just told you. Here's the active site with two metal ions that are a key part of catalysis. We talked about this before when we talked about RNA synthesis. The single-stranded RNA is coming in. It passes the active site. A DNA copy is made. And then the second part of the RT is an RNase H active site. Remember, that's the enzyme that degrades double-stranded molecules. So it will degrade the RNA template that was already used to make DNA. So out comes a single strand of DNA. So it's going to be minus polarity. This is a really slow process. It takes 4 hours to copy one retroviral genome, and it makes a lot of mistakes, about one mistake per 10,000 to 1,000,000 bases. In the particle, the RNA is present in two copies, so it's diploid. You can see the two RNAs here on the left and also in the virus particle. In the particle, the RNA is coated with nucleocapsid protein. The particle also contains about 50 to 100 molecules of reverse transcriptase per virus particle. The two RNAs are held together by base pairing at the very 5' end here. And there is a stem loop here, which has bases complementary to the same stem loop on another RNA, and they base pair in what's called a kissing loop complex, and that holds the two RNAs together. So these are RNAs that have a cap, and they have a poly A tail, and there's the coding sequences for GAG, POL, and envelope. Why are there two RNAs in the particle? It's thought that it makes them resistant to ultraviolet light and ionizing radiation, which cause mutations in viral genomes. There are two copies of all the genes. And so let's say you have two RNAs in the particle, and one of them has a mutation in one of the genes that's lethal. The reverse transcriptase actually flicks back and forth between the two genomes. And so eventually it's going to make a genome that is error-free. So here B and C are normal, but A is mutated in one of the RNAs but not the other. And so the DNA in this case randomly copied the correct base. And so this is thought to build some resistance to mutagenesis. Another interesting feature of the retroviral genome is that there's a tRNA bound near the 5' end. You remember what tRNAs are, right? They are involved in protein synthesis. They're cellular molecules that bring amino acids to the ribosome. But retroviruses have a molecule of tRNA bound to each RNA that's in the particle. And here again at the top are the two RNAs that are stuck together by the kissing loop. And this tRNA, and the bottom here, we've expanded it. You can see the tRNA is not very far from the 5' end of those genomes. And it's binding, it's hybridizing to what's called a primer binding site. Okay, so that tRNA is there bound, and the way it works is shown at the bottom here. Here's the viral RNA. It forms a stem loop. Here's the tRNA, and the tRNA hybridizes with the RNA and insinuates itself into this loop here, this bulge in the middle of the molecule. So every retrovirus has two tRNAs that are hybridized to the RNA. So let's go through the reverse transcription. This will be the last thing that we talk about today, but this is a really amazing procedure. Remember, the virus binds to the cell surface. The particle is internalized, and then the genome is copied to DNA. So the first thing that happens, we're going to now simplify the process. We have the one RNA. We're going to look at one RNA at a time. Here's the tRNA bound to near the 5' end of the RNA. And reverse transcription is going to begin using the tRNA as a primer. The blue is now DNA being made. But it's weird because you make a short piece of DNA and then you reach the end of the molecule. Why is that? Well, here's the story. That the ends of the RNA have repeated sequences. So there's a common little R sequence. And then at the very ends, the 5' and 3', and then there's what's called a U5 and a U3. So when you make DNA, you take the R part and you're making a complementary part, and we're showing that as capital R. The RNA is digested away by RNase H. So now you have a single-stranded bit of DNA, and that big R is going to hybridize with the little r, and then you're going to see the polymerase is going to jump to the 3' end of the genome and keep copying. So here we go. That DNA is hybridizing to big R. The polymerase is now copying this RNA and making a DNA copy. And as the polymerase moves along, it digests the RNA. The RNase H digests the RNA and eventually makes a fully double-stranded, a fully single-stranded minus DNA. Now, the one piece of RNA that's left is called this polypurine tract, and that serves as the primer for the plus strand of RNA. Remember, the first strand of DNA is minus. We're copying the plus RNA. And then when we copy the minus DNA, we get a plus DNA. And that DNA, the second strand, begins even before the first strand is done. All right, let's continue. We finished the first strand now. We've copied the primer binding site, right? And now the synthesis of the second strand proceeds to the tRNA. It can't go any further. The tRNA is cleaved off. But now the primer binding site is complementary on the plus and minus strands. So these two can base pair, and that provides an initiation point for reverse transcriptase to make the plus strand of DNA. And you see that here on this slide. So this is where we just left off. So there's a full minus DNA. We're now making the plus strand DNA. The DNA synthesis goes all the way around until it completely copies that initial minus strand. So now we have a double-stranded DNA copy of the viral RNA. But we have now added sequences to the ends. Remember, there was only R and U3 or R and U5. Now we have three of these. We have U3R, U5, U3R, U5. So it's direct repeat. These are the two LTRs. And that's why there's such a Baroque mechanism, because it has to reconstruct the ends. So here's the RNA, which doesn't have LTRs. It only has RU5. The LTR is U3RU5, U3RU5. So the whole purpose of this jump is to recreate an LTR, because here's U5 and R, and then it picks up a U3. And that gives us the LTR. Why do we need to do that? Well, transcription, as you will see, we don't need to do a quiz. Transcription begins in the LTR. So that double-stranded DNA flanked by LTRs was going to integrate into the host DNA shown in purple. So now we have the provirus. Here's the left-handed LTR, and here's the right-handed LTR. Both LTRs have initiation and termination sequences for transcription. And so the left-handed LTR has an initiation sequence to make mRNA, but it does not begin at the very left end. So you lose the very left sequence of the LTR. And it's similarly, the mRNA terminates short of the end of the LTR. So that's why you have to reconstruct the LTRs when you make DNA, because during transcription, you lose them. And this is going to be the viral genome. And so the genome lacks the full LTR, which is needed to get transcription of the provirus in the host cell. So the next step is integration of this double-stranded DNA into the host cell. That's done by the viral integrase protein. So remember, our virus particle has come in the cell. We've made a DNA copy, a double-stranded DNA copy. That goes into the nucleus, and then the viral integrase protein allows that to integrate into the host cell genome. And so in the end, we have, as I've showed you, integration of a double-stranded DNA into the host cell chromosome, and then transcription to release the genome as an RNA molecule. So we have one DNA made from two RNAs in each particle. There's a strong promoter built in the LTR during reverse transcription. And of course, the provirus DNA directs the host transcription machinery to make mRNAs. And these mRNAs can be encapsulated or translated into viral proteins. But what I find remarkable is that there's no viral DNA replication. There's no DNA polymerase. There's no viral RNA replication. You don't replicate that RNA genome. You take two of them and make one DNA. Now, the integrated provirus, of course, will be duplicated by the host as it duplicates, but there's no viral DNA or RNA replication yet. Obviously, these are very successful viruses. So to summarize that, the virus enters the cell at the plasma membrane. The RNA is converted to DNA, double-stranded circular DNA. It integrates into the host cell genome. Then mRNAs are made by transcription that encode the proteins needed to build new virus particles. Okay, so next time, we're not going to talk about assembly. We're going to talk about reverse transcription part 2, because there's a lot more to talk about. Reverse transcription is really remarkable, okay? And it's being found more and more places. So Dagfinn did not know the opera singers in Santa Fe. They just recorded his lyrics. I guess you had them on some website, right? Right. I'd love to see you on Brian Tyler Cohen's channel. I asked him to invite you. Who got in touch with them? I didn't know. They just sent me the song when they had made it. They're listeners, and they said, we want to make this song for you. They may have said that you wrote it. I don't remember. I'd have to go back to the old emails. Yeah. Steen writes, the University of Maryland story is a very sad example of the human cost of the cuts. I really feel for Xiao Bao Ying, Dr. Simon, who was previously on TWIV. 503 and 512. That's right. Amory, do you think we will have circulation of EV68? Well, it is. It is circulating. It's just not associated with AFM, acute flaccid myelitis, any longer. But it is circulating. Yeah. Logotic, it's not a surprise that Vincent is an EDM fan. He said he's a fan of Miss Monique. Oh my gosh, you remember. That's impressive. Chris Lugo is also good. He looks rather like a Berlin DJ. Me? Wow, thank you. That's a compliment. I just look generic, right? Chris Lugo is a really good DJ. But there are many. There are lots of good DJs. But I did get started with Miss Monique. I like her style very much. FDA approved next spike in May. It's Moderna's latest COVID vaccine. 10 micrograms versus 50, so less reactogenicity. That would be good. Frank Stahl was at the University of Oregon, the Institute of Molecular Biology for many years. The foundational measles and Stahl work was prior to that. There you go. 47 tornadoes is a very bad day, and you might end up fighting a witch. That's great. Well, interesting. Nevitz, I lost smell and taste 18 years ago because of an allergy to benign plant fungus called Alternaria. I was an avid gardener as a kid. Monthly allergy shots reversed it after 4 years. Long haul, but worth it. I'm glad you got it back. That's great. The measles and Stahl experiment. Yep, that's the name of it. Scene writes, I think Rich has made comments to the effect that monkeypox is still the name of the virus. Angela R. is not a fan of ICTV or the new species name. See that Stat News piece. No, it's okay. Don't worry about getting worked up. It's fine. Marco, goodbye. See you next time. We'll be back in 2 weeks. I don't believe it's transmitted. No. Shingles is not transmissible. But if you have the lesions have virus, and so someone who is not immune will get chickenpox, but you have to have close contact. It's not respiratory transmitted like chickenpox virus would be. Recent historical talk by Baltimore Cold Spring Harbor meeting on YouTube was pretty good. So he was at the 50th anniversary of reverse transcriptase. I went out there and did a TWIV with Baltimore, Varmus, and John Coffin. That was cool because they were all around the days of the discovery. For the person asking about shingles, I think the professor's answer could be misinterpreted. You will get sick from shingles, but the virus is a chickenpox virus. Yeah, and you don't acquire that shingles virus from another person. You got it as chickenpox. It remained with you your whole life, and then when it starts to reproduce again later in life, it's shingles. It causes a different disease called shingles. We have not yet found a way to go from protein back to RNA. Well, we can do it in the lab, right? We can take the protein sequence and we can deduce the RNA, and then we can make a DNA copy. But yes, in the cell, that doesn't happen. Seeing how many good insights. Baltimore was mind-blowing to me. I recently got a 68 polio post-translational processing paper. His postulates in the discussion section were crazily accurate. He's a smart guy who thought a lot. And so was Temin. And so was Varmus and many, many people of that era. It was really amazing. What's reverse transcriptase doing in our cells? Well, it's at the ends of our chromosomes. It's part of the telomerase, which keeps the ends from being chewed in during fetal development. And as you get older, the ends because the RT is no longer active when you're born, gets shorter and shorter and shorter, and then you get old. The aging is due to, in part, it getting shorter and shorter and shorter. What else is it doing in our cells? It's copying genes and duplicating them and repurposing them. It can be trouble because these DNAs can move around and integrate in the wrong place and cause problems, right? But it was probably always there. It was probably a relic of the RNA world and then stayed with us since then. It converted the RNA world into a DNA world. Yes, there's TWIV 904 had all the living RT pioneers. That was fun. Does that mean that we can reverse transcriptase for regular PCR? So for regular PCR, if you're looking to amplify a DNA, you don't need to use reverse transcriptase, right? You just need to add your PCR mix. And amplify the DNA. But if you have an RNA virus or an RNA target, you have to use RT to make a DNA copy. EBV question: are people who have autoimmune responses more or less likely to get EBV cancers? If the immune system over-responds, does it prevent cancer or help drive it? I don't know. EBV, good question. So if you already have an autoimmune disease, because EBV can cause cancers, of course, and it can also cause an autoimmune disease. So I'm not sure we know the answer to that. It's a good question. So I have a paper here that says it's not a direct cause of cancer in most individuals with autoimmune diseases, but it can increase the risk of developing some cancers in people with autoimmune diseases, but it's not a direct cause in most people. An old book I read that Baltimore indicated that manganese decreases the effect of reverse transcriptase and magnesium enhances it. Yes, so the two aspartates in the active site each coordinate a magnesium. You can use manganese, but it's less active. So naturally it's using magnesium. Are the prokaryote RTs structurally similar to viral RTs? I know some eukaryotes also have RNA polymerases, but they look nothing like viral polymerases. That's a good question. I don't know. I just don't know the answer. Let's see. Got some scientists here tonight asking me tough questions. They're not identical, which is what I would have guessed. Of course, they have the same catalytic activity, but the structures are very different, as you've said, for these RDRPs in some eukaryotes. Yeah. You would expect that because there's a lot of evolutionary time between them, right? Yes, transposon RTs in archaea and bacteria. That's right. That's right. So the details in these figures are amazing. These are all figures from Principles of Virology that we made over the years. Yep. And these are perfect examples of why we need to fully fund basic research. It must have taken years to figure out all of these mechanisms. Yep. I sent the lyrics to you. I had no idea how they ended up with the performers. I didn't give them to them. I don't know how they got them. And you were credited. Good. I'll see if I can go back and search my email. They have since disappeared after COVID. They don't correspond anymore. But I'm going to try and see how that worked out. They did two. They did two songs, and yours was one of them, obviously. Did reverse transcription put retrovirus sequences into the germline? So, so there are in the germline of many metazoans integrated retroviruses. So these are products of an old retrovirus infection that made it into the germline, right? So the virus has to be able to infect germ cells in order to do that. Not all retroviruses do that. For example, HIV does not infect the germline, fortunately. All right, could you imagine it would pass on to your offsprings and everybody would get HIV? That would be horrible. But some retroviruses do, and so we have many integrated copies of retroviruses that resulted from infections hundreds of thousands of years ago. Okay. And that reverse transcriptase is often active. But there are also reverse transcriptase elements that were not a consequence of retrovirus infection. Those were probably there way before the retroviruses emerged. So probably cells had these reverse transcriptases, including germ cells. And then at some point, it acquired a capsid, probably from the same host cell, and then escaped as a virus. But the reverse transcriptase containing genes were there a long time, and they're in germ cells as well as every other cell in our body. I hope that answers that. Did reverse transcription-like mutations push evolution? Absolutely. Absolutely. So these genes that move around by reverse transcription, what do I mean by that? We're going to talk about that at the next part 2 of it, but the reverse transcriptase encoded in our genome, it's a nuclear enzyme. It remains in the nucleus, and it can copy mRNAs that are produced into DNA, and then those DNAs can go elsewhere and integrate. So you can have gene duplication, and then those duplicated genes can diverge because they no longer are constrained by the original location. They can disrupt genes. So yes, they play a big role in evolution. Do we know how many viruses have integrated into the human genome? Right, so we have 2 kinds of integrations, right? We have endogenous retroviruses, which, as I said, are a product of previous retrovirus infections. And about 8% of the human genome comprises these endogenous retroviruses. We're actually going to go over this in the next mini lecture, but about 8% of our genome. So it's many, because the genome is 3 billion base pairs, right? So 8% is quite a bit. There are a lot of copies, hundreds of copies. And most of them are ancient, right? 100,000 years or more. They're no modern day as far as we can tell. But look, if a retrovirus integrated into our germ cells today, it would take thousands of years before we would see that in the population. So that's why we don't see any modern ones. But that's why the koalas are cool, because they were infected with a retrovirus maybe just a few hundred years ago, and it's that endogenous retrovirus, it's in their germline. It's still spreading in the koala population in Australia, right? Koalas are native to Australia. And so that's there we can see endogenization, right? The process of these retroviral proviruses going into the genome and spreading in the population. Because if you think about it, you have a population of animals which have no of a particular retrovirus. None of them have it in their germline. And then one animal emerges with it integrated in the germline. That has to mate with someone and spread it, and it takes thousands of years for it to propagate. But in koalas, we can watch that happening in real time. It's really, really remarkable. But variation among humans, there are variations because they're diverging from human to human. Once they're in one human's genome, there's a mutation rate per DNA replication cycle, and that makes different mutations from humans to humans. So there are a lot of what we call polymorphisms. But none of them in humans make particles. They're not infectious particles anyway. They're all defective. They have lots of mutations in them that render them non-infectious. Endogenous retroviruses. So jumping genes just need a warm coat to go outside and become a virus. Yes. So here's how I think viruses emerged. So there was an RNA world where you had RNA molecules replicating, self-replicating. They were RNA, and they evolved extensively over time. They, I don't know if protein synthesis evolved in an RNA world. Most likely it did. And we don't know if cells emerged were there RNA-based cells? We don't know. But basically, cells emerged at one point, and then these RNAs went into the cell and acquired a warm coat, a capsid, and out they came as a bona fide virus. When it was a virus, is a mobile genetic element with a capsid. Because there are lots of mobile genetic elements without capsids, like viroids. They're not viruses, they're viroids. And so that's how it happened. And then the conversion to DNA may have happened in the earliest RNA cells, but certainly proteins evolved. The first proteins were probably RNA binding proteins, which evolved into polymerases. RNA-dependent RNA polymerases probably first, and then it doesn't take many changes to make it into a reverse transcriptase. And now you have DNA, and you can make DNA cells, and they can get really big and so forth. So yes, you're right. They just need a warm coat to go outside and become a virus. Mark writes, PDLA protein was virally derived, making it possible to have placental mammals by downregulating maternal T cells. Unfortunately, cancer cells are good at using it too. So the placenta is made possible also by a retroviral envelope protein. So many years ago, before there were placental mammals, an ancestor was infected with a retrovirus. It integrated, and then the glycoprotein gene was used to make the placenta. So the outer layer of cells, the syncytial trophoblasts, are one cell, all fused. Many cells fused together, and that fusion is carried out by a protein called syncytin, which was originally a retroviral envelope protein. So this is another example of how these retroviruses have contributed to evolution of cells, which someone asked before. All right, folks, that's going to do it for another office hours. Great session. I want to thank our moderators, Les, Steph, Vanity Nutrition, and Tom Steinberg. Thank you so much for coming and keeping it civil. Also want to thank everyone for coming here and liking it. Appreciate it, and our discussions are always great. When are we going to be back? So next week I will be in Montreal at ASV. So we will not be doing office hours on the 16th of July. The next office hours will be Wednesday, July 23rd. So we'll have a 2-week break. In the meantime, chill, be safe, everyone. And if you're at ASV, come by and say hello. See you next time, folks. Stay safe. Bye-bye.